Welcome to FYI, the Four-Year Innovation Podcast. This show offers an intellectual discussion on technologically enabled disruption, because investing in innovation starts with understanding it. To learn more, visit arc-invest.com.
ARC Invest is a registered investment advisor focused on investing in disruptive innovation. This podcast is for informational purposes only and should not be relied upon as a basis for investment decisions. It does not constitute, either explicitly or implicitly, any provision of services or products by ARC. All statements made regarding companies or securities are strictly beliefs and points of view held by ARC or podcast guests and are not endorsements or recommendations by ARC to buy, sell, or hold any security. Clients of ARC Investment Management may maintain positions in the securities discussed in this podcast.
Really excited to have you guys on. So Dr. Abudayyeh and Dr. Gootenberg, really exciting, nicest people I know too, but McGovern Institute fellows at MIT. We won't go through total background because it would just take the whole podcast, but researchers at MIT, they're principal investigators there. Omar studied at Harvard Medical School, Harvard MIT Health Sciences and Technology, MD, PhD, completed doctoral work in Feng Zhang's lab, who we've talked about many times on the podcast, the Broad Institute of MIT and Harvard, a lot of research on CRISPR enzymes, which are also one of our favorite topics to talk about on this podcast. We love genome editing, so that comes up a lot as well. But was also recognized in 2018 in Forbes 30 Under 30 in Science and Healthcare and Business Insider 30 Under 30. So a lot to talk about there. And then very quickly, Dr. Gootenberg earned his bachelor's degree in mathematics and biological engineering at MIT, PhD in systems biology from Harvard, and conducted research also with Feng Zhang at the McGovern Institute and Broad Institute of MIT and Harvard, and focused on molecular technologies for targeting and sensing disease states, crossing disciplines by utilizing novel computational techniques, microbiology, biochemistry, molecular biology, for new CRISPR tools, which are some of our favorites, including Cas12 and Cas13, which we've talked about before, and I'm sure we'll get into it more now too. And Dr. Gootenberg was also in the 30 Under 30 and Business Insider's 30 and Under. So sorry, that was a mouthful, but so many amazing accomplishments. So those were just a few of them, but I don't want to make you guys blush anymore, but just wanted to get some of those out there. But thanks for coming on.
Yeah, no, thanks for having us on. No, I love the opportunity to talk.
Cool. So I guess we can start in so many different places, but maybe my introduction just focused on some of the, huge accolades, but maybe do you guys just want to talk for a second just about your background and how you got to where you are today?
Yeah, no. Omar, why don't you start?
Sure. So, Jonathan and I have been working together now for a number of years. We actually first met in college at MIT, where we did a biodesign course together, and I guess became science soulmates after that course and sort of crossed paths again in different labs. We crossed paths again a couple of years later in Ed Boyden's lab, and then both landed in Feng Zhang's lab as PhD students. And I would say our science sort of romance didn't rekindle again for maybe another year in the lab when we started working really close together after a year of sort of starting there. And, our early sort of collaboration was on a lot of the new CRISPR stuff. So, Cas9 was just picking up around that time. And we asked a really interesting question, which is Cas9 really the only thing out there? In other spaces in molecular biology, for example, molecular cloning, where you might want to copy a gene for the purpose of, doing gene therapy or making vectors that can express genes. And there are thousands of different restriction enzymes. And so we were wondering, was Cas9 just the first example that was found for sort of CRISPR systems? And so we embarked on a journey of 6 or 7 years of discovering all sorts of new CRISPR systems, Cas12, Cas13, things that target RNA instead of DNA for reversible gene therapy, stuff that was useful for CRISPR diagnostics, not just gene therapy. We started a number of companies in both the CRISPR diagnostics space as well as the gene therapy space, some with Feng as well. And, a few years ago, we actually started our lab together at MIT to sort of keep churning out new molecular tools for gene editing and cell engineering. And we're happy to chat more about where that's going. So I'll pass it back to Jonathan to clarify any holes in the story.
No, that's a pretty good, I'd say, summary. I mean, I think just to take it from a different tack, I think both of us, before we started working together, we were very interested in tool development because it's an old trope, but scientific research is, driven by tools. And I remember when I was, I loved biology when I was a kid and got to work in some labs, and that was fun. But I think so often you hit this kind of philosophical question, which is, what do you work on? And one nice thing about tools is that working on methods, I mean, CRISPR obviously is probably the prime example right now in people's heads, but methods have been in biology as long as biology has been around, is that if you develop something that really is useful, it lifts everybody. So anyone can take that and use that. And that's a nice thing about molecular tools in general, because, CRISPR, the Cas9 enzyme in the ribonucleoprotein complex, it's incredibly complex. I don't think I could build that myself, but it's really easy because you just put that DNA into a bacteria or a human cell or whatever, and it does all the work for you. So it means that when you work on these methods and tools, that is incredibly satisfying to be able to make something that can help everybody. And so Omar and I were, I think, inspired by that before we started working in Fung's lab. When we were in Ed's lab, we were working on ways to think about how to use molecular tools to sequence proteins and a lot of crazy ideas. But I think that we were both drawn to the concept that you could take something from nature or engineer something de novo or combine those and really use that to have an amazing benefit in basic biology, in therapeutics, and diagnostics. And that's, I think, the core of really what we are still inspired to do. And Omar mentioned that we started out in kind of genome engineering, but it's been really amazing to see how kind of the different fields of really cutting edge science have been evolving even while we've been in Boston. And I was co-advised by Aviv Regev, and so I had a lot of exposure to this idea of kind of single cell and cellular profiling, this idea that we can actually look at heterogeneity in disease in the body at the single cell level. And now it's becoming clear that these two fields of genome editing and single cell analysis are really coming under this incredible large umbrella of how do we profile cells and then engineer cells. And I think that's really how we view our, kind of overall lab goal, which is how do we develop tools to make the next era of cellular engineering really possible, so we can control cells as medicines and as diagnostics to really have entirely new ways to attack questions of biology and questions of health.
Yeah, I think that's really good context. It's really helpful. And also, I want to find my science soulmate. So, hopefully that happens soon. But it's interesting you mentioned sequencing proteins and how crazy it was. And the cool thing is that you guys are so forward thinking, that was crazy at the time and now is becoming, a little bit less crazy. So there's so many different places that we can start and go through here. But, one of the things that would be interesting to talk about is, as you mentioned, diagnostics. So we do a lot of these podcasts. We talk a lot about DNA editing. So I'd love to get into RNA editing, which you guys are obviously at the forefront. But diagnostics is something we don't talk about as much as well. So maybe we can just go into a little bit about diagnostics. Obviously, we can touch on Sherlock. The creation of detecting human disease is really interesting, and hunting for, these CRISPR tools that can hopefully treat disease and maybe even those related to aging. So maybe you guys can touch on sort of the idea of creating new diagnostic tools using CRISPR.
It's an interesting concept, right? Because a lot of the excitement was around CRISPR for gene therapy and gene editing. And what's amazing is the precise ability of these enzymes to precisely find their target site in the genome should intuitively make sense that, they'd be really good at finding, sequences that are, not endogenous to the human genome if they have this search function. And so I think it was kind of like this concept of like, well, instead of like trying to find a sequence in the human genome and cut it, let's find something else, like a virus that shouldn't be there. And then instead of cutting it, like report on the presence of that. And so, we stumbled just coincidentally on some enzymes that were more attuned for being able to do diagnostics rather than editing, just because of their natural sort of biochemical functions. And we realized this would be the perfect system to showcase these diagnostic potentials. And I think, what's really exciting is beyond whether, it's CRISPR or some other chemistry, the concept of like, what's the smart home of, the next decade or 2 decades, right? And, we're starting to get all these, things in our house, the Internet of Things, like objects that can listen all the time and tell you the weather when you wake up in the morning, or turn your lights to like a million different colors. And, that's really cool, but wouldn't it also be cool to have like an automated sort of clinic in your home as well? That could, if you feel sick in the middle of the night, you could just swab yourself and it tells you, oh, you have like a cold, or, oh man, it's pneumonia, go to the hospital. It would be, cool then if that result could then, be used for like telehealth. So you don't even have to go anywhere, but you just hop on the phone and someone tells you what to do. And so I think we're heading in that direction as molecular biology becomes simpler and simpler, and biology itself becomes more and more programmable. I think we're starting to see this with like Moderna, right? Being able to make a vaccine in less than a year, and talking about programming ourselves for new, COVID updates and, other bugs, even like, flu. We're starting to see this acceleration in other spaces. And I think the concept of bringing this type of really technical molecular biology into every person's home to, give you complete information about yourself and your health, and when things maybe go wrong, would be really exciting, and it doesn't exist yet. And so that's kind of the concept behind CRISPR diagnostics.
Cool. Jonathan, anything you want to jump in?
Yeah. So I guess to take a different tack on CRISPR diagnostics, just kind of as a background for how it works, that when these enzymes, a subclass of these enzymes, they identify sequences, they become activated, and then they cut up other things in the test tube, and we can use that as a diagnostic because we can detect that cutting activity. And I want to actually use this for a different teaching, which is that you never know what you'll find. So when we first found this property of kind of activation of cutting things up, we called it collateral cleavage. We found it on the enzyme Cas13, and we had no idea that this enzyme had this property. It's actually, we always had assumed up to the point that we did the experiments, that we assumed it worked exactly like Cas9, which is that Cas9, one metaphor is, it's this programmable pair of scissors where you target it somewhere and it cuts right there. And we thought that would work the same way. But the crazy thing is, essentially structurally, the part that identifies in Cas13 and the scissors are in different places. So you identify and you activate somewhere else. And we couldn't have known that beforehand. So I think all those CRISPR diagnostic applications that Omar outlined, these kind of really amazing blue sky opportunities, we had no idea that these enzymes had this until we just characterized them, and we found it by happenstance. So it really speaks to the fact that we need to continue to just explore natural diversity, kind of interesting mechanisms, and that we can probably end up turning a lot of these things unexpectedly into tools just by following our curiosity. So that's my CRISPR diagnostics spiel.
That was a really good breakdown of exactly how it works, because I think that's definitely been a challenge for some people to understand. I think it'd be cool if you could just to talk about fluorescence and how it works when there's actually something that's detected.
Yeah. So basically we have this enzyme and it's going around searching for this sequence, and it finds the sequence that we want it to find. Let's say the SARS-CoV-2 genome, right? It becomes activated, and then it starts cutting things in the solution, pieces of RNA in the solution. So what we do is we have a different molecule that we spike in, and it consists of a piece of RNA, and it's a little bit like a dumbbell, but it's an uneven dumbbell. It's not symmetric. So on one end is a fluorophore, it glows, and we can detect that. But on the other end is something called a quencher. And that's a molecule that actually blocks the fluorescence of that fluorophore. It quenches it. So pretty apt name. And this means that when those two pieces are together on that dumbbell, it's dark. You can't detect it. Now, when that Cas13 that's become activated, and Cas12 also has this property on DNA, but yeah, this is a bit of an aside, it cuts that RNA and separates and breaks that dumbbell. And those two pieces, the fluorophore and the quencher, are then separated. And that actually means that the quencher can no longer quench the fluorophore, and the fluorophore glows. And then we can detect that by normal fluorescence, like a easy LED in a detector and in a very portable way. And it means that we can actually use this to make a glowing signal that can even, in some cases, people have used CRISPR diagnostics to see things by eye, which is very cool. We can also, using a bit of a different hack, make it so we have two molecules that we have together and then that cutting separates them. And we can actually detect it on what's called a lateral flow strip, which is one common version of those is a pregnancy test. So it's actually like a pregnancy test format that you can use for things like, COVID. We've actually demonstrated that lateral flow strip readout for COVID, which means that you actually don't need any instrumentation for readout at all. It's very easy to use. So it's a very modular technology, and it means that people have used this for actually a lot of different flexible things, like Jim Collins, who we've collaborated with a ton, and he was early on one of the co-authors, and his lab was integral in starting the Sherlock work, made it so it's actually electronics. So you can gate electronics using this CRISPR detection. And he had a recent paper where they actually integrated this technology into a face mask. So you can use CRISPR diagnostics and CRISPR detection in a wearable, and then that could trigger something like an electronic signal or a persistent sensor. So it's very flexible and very useful in a lot of different circumstances. And that's one reason I think why CRISPR diagnostics is having such an impact.
That makes a lot of sense. And obviously, we've seen that with COVID, it's been an interesting potential use case. Omar, just want to throw it back to you if there's anything you want to add on that.
I think what would be really cool, we've obviously spent many years optimizing the chemistry and how to, make these things as portable as possible. But I think really the next step is more on the device side. And I think, it's interesting when biology and the chemistry gets to a certain point, it really becomes more about like, how do you interface these things with people and consumers? And so we've been spending a lot of time looking at, how to build a thing like an Apple HomePod, for example, that could, house the whole system. And, you basically just swab yourself and put it in, and it's automated and can run in, sort of any setting. And I think when we think about more of like, how does this work for consumers? I think the interesting things are, COVID is one thing, but like, respiratory panels, like things that, that tell you whether you should go to the hospital or not, like flu versus something more serious like pneumonia, or something that's just the regular cold, or for kids RSV versus CMV versus other different respiratory viruses. So thinking through like the menu and like what panels sort of make most sense, probably most likely in the infectious disease space, I think is what'll be most interesting. But obviously we interface with clinicians and advisors to really work through these things. But those are our current thoughts. Yeah.
I think the device side is super interesting. How far away do you think we are from actually having that? Because, there are devices like the Oura Ring or others where you can look at, sometimes some symptoms that are early and it can help with detection. I think there's been some studies on the Oura Ring specifically that have shown, that there's been some sort of early symptoms of COVID. So they've been able to use that. But just curious how far away you think we are from like actually having these type of devices at home where we can use them as diagnostics.
Yeah, I think that, probably sooner than you might anticipate. We're actively working to translate some of these technologies and chemistries into basically a box that you can have a different menu of tests. So that's currently undergoing a lot of testing optimization, but we're hoping that it'll be out quite soon. So it's something that I think consumers will be able to access sooner rather than later, probably hopefully within the next year or two.
That's awesome. At the beginning of COVID, we looked at how much employers would save if employees didn't show up when they had any signs or symptoms of being sick. And we found, I can't remember the number offhand, but it was quite substantial. And so if you just invest in these technologies, then it's really possible to, potentially actually save money in the long run, because then the sort of infection chain ends and you're able to alert when you wouldn't go to work. And obviously your diagnosis can be used for so many different reasons, but having it at home is just, beneficial in so many ways. And I guess employees or employers saving money is not the first that comes to mind for you guys probably, but it is a substantial amount of money. So it's probably an interesting avenue to think about too. But maybe now we can switch to RNA, because I think even for me, I didn't realize how impactful RNA is, in gene editing. When you think about, traditional gene editing or DNA editing, firstly, we obviously need the guide RNA. So that makes it pretty central. But also there's, a few parallels to RNA interference with DNA editing. And then also Dr. Jennifer Doudna is a biochemist that specializes in RNA. And Emmanuel Charpentier, who we know both of them won, the Nobel Prize for CRISPR, was really fascinated with tracer RNA. So anyway, all this to say that RNA is obviously very crucial to the gene editing story. So would love to kind of start talking about RNA. And obviously you two have become very influential in RNA, given the Cas7, Cas11. I think that was only a couple weeks ago that the paper came out. So first of all, congratulations. Really exciting stuff. And I'd be curious to know, so in the lab, you're trying to figure this out. What was kind of like the eureka moment where you're like, oh my God, we just did this?
Yeah, I mean, there's a couple of them, but I think that with these systems, when you're kind of working on these CRISPR systems, it's always nice to show the first activity, which really like showing we put this into a system and it worked. So for us, I feel like, or at least for me, I think Omar probably has a, of course, a different perspective, but the eureka moment is when you put this into a bacteria and then you try to put in a virus against that bacteria with an RNA genome, and you show that it cuts that virus's genome and it protects. And then you're, wow, this works. Maybe it doesn't work in mammalian cells. That might be another eureka moment, actually, when we got it to work in mammalian cells. But it's an enzyme that actually does this programmed cutting. And I think that's always cool to see that it's an actual functional piece of protein that's doing what you want. At least that's, when I thought it. I don't know if Omar, maybe you did a different eureka moment.
This most recent enzyme Cas11, as for people who don't know, there's most people think of CRISPR as DNA targeting, and we've spent a lot of time also finding sort of RNA targeting systems. And I don't know if everyone attending here is a biologist, so I want to explain that in a little bit more detail. But DNA, of course, is sort of this permanent genetic code, right? That we all have that tells cells how to differentiate and form tissues and run all our processes. And RNA are pieces of the genome that are expressed that actually can turn into proteins. And RNA is much more transient, right? It can turn over in a matter of days. It's not permanent. So when you edit your DNA, you make permanent gene edits, and that's great for, one and done sort of gene therapies. But if you edit RNA, it can actually be reversible and turn over. And so, we had a lot of excitement in developing RNA targeting CRISPR systems for the purpose of being able to do sort of reversible gene therapies. Think of it as like a pill. And when you take that pill, you can edit a base in your genome that treats a disease, and you can always stop taking that pill, and that can get around any potential side effects of permanent gene editing or potential off targets or whatever. And so, I think early on in the RNA targeting world, we had our first eureka moment. This must have been like 2016, 2015. We were trying really hard to find a CRISPR enzyme that would actually cut RNA. And, we probably screened through dozens and dozens of different proteins from all sorts of random bacteria. And eventually, I think a late night, like in the radiation room where we like image our gels, we actually were able to finally see a smear on the gel that indicated the RNA was being cut. It was actually really weird at the time because we were looking for precise bands that indicate precise cutting, and instead we were getting a smear because the RNA was being degraded. We found out later that these RNA targeting systems, these Cas13s, actually don't make precise cuts. They actually recognize a target and then cut the RNA to degradation, which is made sense of that discovery later. But it, I think goes to show that not all discoveries at first really make sense, and it can take a whole nother year just to sort through all the data and put it all together. And what's interesting is this Cas11 paper we published like a month ago was the culmination of all that work, because we finally found a CRISPR enzyme system that makes a precise cut rather than having this sort of collateral activity that blows up the cell, which is why we sort of show in the paper that makes for more safer RNA targeting therapies. As an example, if you want to use these systems to target COVID, because COVID's an RNA virus, you can't use Cas9 to target it because Cas9 doesn't cut RNA. Cas11 would be really great because you could precisely design it to cut the virus when it gets into your cells, and you could clear the virus better as a therapeutic option. And so these are the types of things these tools and technologies can be useful for.
Right. That's actually really helpful and good context. And no, everyone is not a biologist. So really appreciate the extra explanation, because I'm sure that helped a lot of people. So, when I think about RNA editing, I love the idea that it's transient in the sense that it's reversible, because, as we know, and we've seen in trials, sometimes we get news that is disconcerting a little bit with off-target edits. Like, very recently with Allogene, they had a patient with their edited CAR T who had a chromosomal abnormality. We await further data, but I think, hearing that is nerve-wracking, and we want to ensure that there are no off-target edits. And, I think gene editing will get better and better with different iterations, but it is interesting to have an option to do RNA editing, which is transient, but the nature of it being transient is also, difficult, because typically that would mean that you need to redose. So, maybe help me understand this. So with RNA, I think about the half-life and the idea that, we're constantly getting some new RNA. And so I wonder, does the RNA need to kind of linger around and be present in the body for, the therapy to be continuous. And then also I'm curious about sort of your ideas versus, a catalytic versus more of a stoichiometric therapeutic, because I imagine that, RNA would be more sort of stoichiometric. So just curious if there are any thoughts there and sort of like advantages or disadvantages.
Yeah, explaining catalytic versus stoichiometric. No, I mean, that's actually a really good point. But to kind of take it back to RNA versus DNA. So CRISPR is really amazing because it gives us the capacity to program these enzymes to go to anywhere in the genome and edit it based on rules that we understand. Base pairing, A's and T's, C's and G's. With RNA, it's A's and U's, but same idea. But, the idea that we are only currently using that to target mostly genetic diseases is because I think that there's a lot of cases where you don't want this to be permanent, right? This programmability. And we're not just talking about changing the letters. We're also talking about reducing RNAs and upregulating RNAs. But the idea that you can actually now do this means that you can start to hit places in the regulation of the cell that actually allow you to have a lot of potentially very unique control over cell state. And here's a small vignette about that. You don't necessarily have to knock something down and keep knocking it down to have an effect. There's the other school of thought, which is that you can knock something down to create temporarily, to create a permanent effect. And that effect could be turning one cell type into another. So there's a lot of interesting work on transdifferentiation where you can knock down a certain transcript, in one that's commonly used is PTBP1, to actually change the identity of a cell from a non-neuronal cell to a neuronal cell. And this, you only have to transiently knock it down, but it's actually a permanent change. And people have done this actually with RNA targeting CRISPR enzymes. There's a small footnote to that, which is now people are looking into the biology of that. And there was recently a paper that raised a lot of questions about that transdifferentiation work. But the concept of being able to cause a permanent change in a cell's identity to turn it from one cell type to another, or in a different case, to knock down a transcript to cause proliferation of a cell to regenerate your liver, could be a very unique approach using RNA modulation to create a permanent change. There's also instances where you don't want something to be permanent at all. So people have used modulation of RNA via CRISPR to knock down certain potassium channels and to actually regulate and reduce pain. So to use it as an analgesic to actually be able to eliminate pain. And people are using different CRISPR tools for this. But this obviously you wouldn't want to be permanent and you would want to be able to redose, but it would be a non-addictive pain medicine. So being able to think about those potential cases, I think, shows a lot of the utility. And at the other end of the spectrum, you could deliver an RNA targeting enzyme and have it permanently expressed and actually be able to knock something down for a long term. And you could use that to repress the levels for a long period. This would be as an alternative to RNAi, where you may not want to be able to use the natural RNAi machinery for safety reasons, which have been put into question. So really, RNA targeting CRISPR approaches encompass both permanent and temporary approaches to creating changes in cells, causing cells to do different things, targeting genetic diseases. And that's why I think they're so exciting, because there's just so many things you can do with them as a platform.
That's awesome, because you touched on a few things. Wait, I want to throw it back to Omar. I know you said you want to give Jonathan the hard ones, but anything you want to add on that one?
Yeah, well, maybe I'll just tackle the first question you had, which is, what is RNA editing good for? And I think, I mentioned one of them, which is targeting viruses, right? Because you don't need to permanently express this technology to knock down the virus, right? You just need to deliver it, probably deliver it as mRNA and synthetic guides, much like our vaccines are delivered. And, you let it, incubate in the cells for a few days, or maybe you need to redose a bit until the virus load goes down. So I think infectious disease is a really good application for these types of systems. And the others are like what Jonathan was saying, which is knocking a cell into a new state. There are a lot of examples where you can knock something down to convert and transdifferentiate cells, for example. Maybe you want to make neurons in the brain and you can use RNA knockdown for that. You don't need to permanently express it. Maybe you want to regenerate cells post post-liver injury or a heart attack. And RNA knockdown can do that too. So I think there actually are a lot of short-term modulation opportunities that are not being met with current technologies. And I think programmable RNA CRISPR technologies offer a really cool solution to that. And so hopefully in the future, we'll have that.
Yeah, that certainly would have been helpful a year and a half ago. So, so, get working on it, guys. But no, you guys touched on a few really great points. So one was sort of this differentiation between other modalities like antisense oligonucleotides and RNA interference. And the other one that I think you guys touched on that, would love to talk a little bit more about is the safety. So, one thing that's on kind of the forefront of my mind is this weekend, Ionis and Biogen presented some data for their SOD1 ALS trial. Unfortunately, it did not show efficacy when compared to placebo. Obviously, we saw some change in the neurofilament, but I think we're seeing with ASOs at least that there's some difference in the pharmacodynamics, but we're not really seeing it in the efficacy. So I don't know, maybe this is timing, maybe this is dosing. Some people think this is still enough to get approved. That's maybe too political to talk about right here, but this feels a little bit like deja vu for me, at least with Aduhelm or aducanumab. So I'm curious, how do you guys think about the space in terms of like RNAi, ASOs, and obviously, you did a great job about talking a little bit about, what RNA editing specifically would be used for, but do you kind of see these as all sort of serving a lot of the same indications, or do you see these as kind of differing? And is one of the main differentiations going to be safety?
Yeah, so just on the safety side, I think it's always hard to kind of think about that because obviously it's a long road and there's many things you can't anticipate. I think that when you're developing these medicines, things may come about during production of the vector or, there's just a paper from Bev Davidson's lab that they saw toxicity with an microRNA AAV, which I saw that and I was like, well, I would have assumed this because RNAi has been shown to have, in a lot of cases, off targets, but they actually found out it was the vector itself being actually the ITRs were causing some transcription of something that was toxic to neurons. It's a little bit inside baseball, but I think the take home is it's very hard to predict when you're making these medicines where things might go wrong. But on the safety standpoint, I like the aspect of CRISPR-based tools, especially Cas711, because they're completely orthogonal to the biology of the body. When you take something like RNAi and you go through and actually dose a patient with that, you're co-opting natural machinery, which can take factors out from things like microRNA processing. And Mark Kay has shown for a lot of different RNAi therapeutics that they actually cause liver toxicity through some of these different modalities. So I think that's one thing that I like about being orthogonal. The flip side of that is that sometimes you have to express a foreign protein, which can also potentially cause an immune effect. So everything has a trade-off. What I do like about Cas711 and Cas13 and these RNA targeting CRISPR systems is that we can engineer them, which we can't with RNAi. We can actually use protein engineering technologies to put them into different places in the cell, whether it's the nucleus or the cytoplasm, be able to control them, to be able to actually make them triggerable by things like small molecules. And that allows us an extra layer of safety, of tunability that you can't necessarily get with something like ASO or RNAi. I also like the permanence. With ASO, you're not going to be able to actually deliver with an AAV and have that stick around for a long time. And we can actually continue to express this. And it's an enzyme. It'll cut multiple times. ASOs, they'll bind and they can recruit things like RNase H, but they probably don't have that same turnover. So I think there's a lot of different ways that they can be used, but I think it's very neat that they can overlap in certain problem sets. Of course, with these CRISPR targeting, these RNA targeting CRISPR systems, we can also put things on them to actually modify the bases of RNA, to actually change the epitranscriptomics of RNA, which is additional marks on the RNA. So it gives you a lot more optionality there where we can actually start to tune the biology much more, actually, I would say, precisely. So I think that is one of the other large advantages of these CRISPR systems versus things like ASOs and RNAi.
Omar, I'm throwing it to you if you want to add anything.
I think what, Jonathan touched on the most important points. I mean, I think, the preciseness of these systems and them being exogenous makes it much easier to build upon them, right? A lot of times you may want to actually use these systems to recruit new enzymes like a base editor, which is going to be tough to do with like RNAi or just, antisense oligos. And I think, Jonathan touched on the points of specificity and toxicity as well. So I think, that obviously highlights some of the advantages of the CRISPR-based approaches.
That's actually an interesting point. Like these increased sort of functionality, like base editing or prime editing, those can't really be applied obviously to ASOs or to other, forms, but to RNA editing they can. So, the differences will only get that much more grave as we kind of continue to build out the RNA editing sphere, which is pretty exciting. How do you think people are going to kind of perceive the safety when you compare this relative to like small molecules?
I think that's, a great question. We actually, as, with our drug development hats on, talk about this a lot. And, with, when you think about like working with FDA and the types of data you need to submit, I mean, obviously with small molecules, you don't go nearly as far with the lengths that you have to go to for like gene editing technologies, where you have to look, as deep as possible and find every possible off target and, characterize all the guide RNAs you're using and so on. And, look at like, effects of like RNA expression and so I think, with these, systems, we're actually doing much better characterization than you might get with a small molecule. And so I think as with anything, you can have, designs that are, really not specific, and you can also engineer designs that are really, really good and specific. And so, I think because we're characterizing these things and we're looking at it and we're screening all sorts of modifications that can make these things as specific as possible, they're getting really good now. And, I know there's concerns about off targets in both the DNA and the RNA space, but it's surprising how well a lot of the companies have been able to come in the past like 7 to 8 years in getting these systems to be exquisitely specific. And because we actually characterize the specificity, we know what we're getting out of the systems. And I think that goes way beyond what you get in the small molecule world.
Cool. That's awesome, because that was an answer I just was not really anticipating. Because I think like public perception would be, hey, it's a small molecule, it's been around for forever, the safety profile is there. But it's super interesting to hear that, based on the testing that you're doing, it actually may be that this is going to have an even better safety profile.
Yeah, well,
really
sorry, to clarify, to make sure I'm not saying that approved drugs are not safe. Obviously, they go through clinical trials and are shown to be safe. I guess I'm more talking at the molecular specificity. Even though the small molecule is safe at a human level perspective, these small molecules are probably modulating a lot of, different things in the cell. They may not be wreaking havoc, but they're still technically off targets. And I think that's really interesting. And I can't remember, I think there was a paper that actually looked at a lot of approved small molecules and how they actually hit targets that we never intended them to hit. So that's the point I'm trying to make more. And I think, obviously the first, phase 1 results have come out for a lot of the current gene editing approaches. I think it's still, touch and go. There was data from Allogene, I think last week, that showed, there could be really, really rare events that might have an effect. And I think time will tell whether this is an issue for the CRISPR-based cell therapies as well. But I think, I can say at least, the companies are doing the work, and there's so many cool engineered specificity versions of these tools, as well as ways to read out specificity, have been developed. So,
Jonathan, anything to add on that one?
No, I think that's pretty good.
Don't mess with perfection. I got you. Okay. So I think, I'm curious. So I've heard Emmanuel Charpentier say that, or I might have read this. I'm reading Editing Humanity, as you guys know. So it might have been in that book that I saw this, but she talked about how delivery is a bigger bottleneck than the CRISPR mechanism itself. So it's funny, we talk about safety, but maybe it's not safety. Maybe it's just actually the delivery is really challenging. So, what's kind of, we've touched on delivery throughout kind of the conversation, but do you guys want to add anything in terms of delivery? Obviously, the FDA met already to discuss, toxicity concerns with AAVs. So, how are you guys thinking about delivery? And, do you think delivery will be, easier, harder, different for RNA editing versus DNA editing?
Yeah, I mean, it's different. You need to pick the right tool for the right job. But overall, yes, I mean, delivery is key. I wouldn't say it's like more important than the CRISPR enzyme or not. There are two parts that you both need. You need the scissors or the way to cut or edit the molecule you want, and you need to get it to the place you want. So I think that delivery, obviously, there have been immense inroads into now non-viral methods. The Moderna LNP, well, the LNP that's in the Moderna vaccine, I should say, and the abundance of these different ways to get things into cells have really, I think, catalyzed a lot of ongoing work that was already really fantastic. And Intellia's LNP data from their Novartis licensed LNP was really inspiring. So the idea that we can now use a lot of non-viral approaches, I think, has really upended a lot of the work. So people think it's not just viruses, it's not just AAV. I remember in 2014, 2015, when we were in Fung's lab and we were working on this smaller version of Cas9 from a bacteria called Staphylococcus aureus that could be packaged in AAV, we thought that this would be like the large enabling thing, that you could now put this into an AAV. But as you can see, there's many other ways to do delivery. Now, I think delivery still has a lot of challenges. It's very hard to get to places that aren't the liver still. And I think that there's a lot of fantastic work being done. And we'll see a lot of creative approaches that mine things like natural biology to either find new viruses or to adorn non-viral nanoparticles with things that look like natural ways to put things into the body that'll help with this problem. I think also specificity of delivery is a big question. So it couples it with toxicity. So maybe you can get to certain cells, you get out of the liver, but there are also other cells you're getting to that you don't necessarily want to get to. I think that can be sometimes a question, especially if you're delivering something that could be very potentially changing of a cell state, like with RNA targeting. And I think that there's a lot of interesting work that we're thinking about in our lab about how do you control a payload. So even if you get into the wrong cell type via your delivery method, then you can do logic and understand, well, this isn't the cell with the expression profile that we want. That's not expressed here. So I'd say there's still a lot of challenges. When we think about RNA delivery, we mentioned before short-term versus long-term. So if you want to use short-term, we can use an LNP and have it around for much less. If you want to have long-term, we can use one of these viral vectors like an AAV and have it more persistent. So to go back to, you want the right tool for the right job, and it really depends on your indication. But it's really inspiring to see that we have this ever-expanding menu of ways to get into cells in the body that I think will probably just continue to grow and grow.
So, as you're talking, it starts to get me thinking just about how we detect off-target edits. And as you mentioned, we need the right tools. So I know when you use the word tools, you were talking more about delivery tools, but it started to get me to think about checking and having the right tools for off-target edit detection. And so I'd be curious to know your thoughts. I mean, ARC has done quite a bit of work, and a shout out to Simon Barnett, who's my co-analyst, who has done a lot of work on this, but on the difference between sort of short read and long read sequencing. And just curious if you guys had any thoughts, about what are sort of the right tools to get the right information of if there are any off-target edits.
Yeah, no, I think it's a great point. I mean, and I'm sure, most people probably use a combination of all the technologies out there. So not to get into the jargon, but, the short read technologies like, Illumina allow for really, precise, either like sort of semi-high coverage, panning of the genome for, small edits, as well as targeted off-target sequencing. The bigger issue, I think, which maybe goes underappreciated, as looking for large off targets, like things that cause chromosomal recombinations or large deletions, which obviously typical short read sequencing like Illumina can catch. That's actually probably what affected the Allogene trial from last week that I was referencing, where you got some clone that grew up probably because of a large deletion or rearrangement. So I think, that's what you're mentioning, where you have to go into either like nanopore sequencing or, God forbid, pack bio sequencing, which is even can do really long reads, but takes a long time. And so I think anyone would have to have like a whole pipeline basically to look at these things and analyze them. And what's actually really cool, which I just thought of, Keith Jung, who spent a lot of time, he's a professor at MGH, founder of a lot of gene editing companies in the area, actually started a company to literally look at off targets for people. I'm trying to remember the name. I think it's like SecurityX. Do you remember, Jonathan? I think it's SecurityX. Yeah. Basically, it's like a pay for, I don't know what to call it. pay for service model or whatever, but like companies can literally pay them to get like an off-target analysis done for their like CRISPR drug or whatever. So that's this is obviously still an early time for the field, and these things are being built.
Yeah, it's an interesting time, and I think there's so much unknown. So you guys are used to that in terms of the lab of working through these unknowns. But I think to the investor community, it's interesting investing in biotech and having the unknowns and still feeling very passionate about the companies and about the science. So it's an exciting time for sure. I think we've touched on this as well, but with the pandemic and how quickly we were able to make vaccines, I think that this is just going to be true for RNA therapeutics, right? So isn't the iteration time for like an RNA therapeutic development, won't that be much shorter than for a small molecule, let's say?
Yeah, I mean, I think that it's obviously case dependent because, once you have the platform, you have to start to worry about the biology. So if you're delivering an RNA therapeutic with an LNP that might not be toxic itself, that approach could potentially have no, off targets or toxicity. But the actual mRNA you choose for your disease, depending on what it is, that could have a lot of considerations. So I think that we'll see some things that move very rapidly, but I'm sure that there will be a lot of unexpected biology. And even if you can knock the gene down or edit the gene or do whatever, sometimes the biology doesn't work on the efficacy side. And we just can't anticipate that.
So I think, we have a 5 to 10 year time horizon. So we think of things as, we don't need sort of the immediate result, but we believe in the companies and sort of the management teams. And we sort of, like to have the vision and the forethought to think sort of futuristically as you guys do, because you're developing these therapeutics. So what's kind of coming down the pipe. So I guess I'd be curious, do you guys have any sort of thought about like what is going to be in 5 to 10 years? Like, are we going to be still talking about Cas9 IP? Are we going to be talking about RNA editing a lot more? Are we going to be talking a lot more about prime editing? What do you kind of think is going to be the main conversation in 5 to 10 years?
Honestly, given how fast the field moves, it'll probably be a technology that's not even invented yet or thought of. But, remind us like, the original CRISPR nucleases are just, coming on their maybe 9-year anniversary of being published. And, God knows where we'll be in 10 years. But, I think in terms of where we're going, I mean, one, maybe hot take I'll put out there is we're getting so much focused on, more and more precise, editing. And, it's funny because when you take a step back and look at some diseases like cystic fibrosis, for example, which has 1,800 mutations, it's like, how are you going to make, 1,800 different prime drugs or 1,800 different, Cas9 edited drugs? It's a lot of work for one company or even, multiple companies to do. And so, it's when we think about like what is needed for some diseases, and a lot of diseases actually have dozens or hundreds of mutations, and there isn't like, one mutation that's dominating, right? So if you only develop the most predominant one, you're still probably leaving a lot of patients at the table. And so, one thing I think that's still left for the field is obviously programmable gene integration, which, instead of saying you're going to target like a mutation, you go broader and say, I'm just going to replace an entire gene. And, there you can actually hit all the mutations at once, and you can make a single drug that affects, all the patients. And so I think, probably we'll be talking about that sooner than 10 years. But, just, a hot take for, the balancing of like making the most precise, editing technology versus, things that can maybe do larger types of things that can treat more people and are more practical to actually develop drugs for, I think is one thing we'll see the field turn towards. In 10 years, I mean, I think, we'll probably see more work towards, I think, in vivo cell engineering. So moving away from just genetic engineering tools, I think, obviously there was this huge, sort of Cambrian, evolution almost of gene editing tools, right? We sequenced the first human genome, we sequenced many human genomes. We needed tools to start, manipulating the insights from those things. And we built a genetic engineering toolbox. And I think for the next decade, it's like we're now sequencing cells, right? Single cell RNA sequencing, single cell epigenome sequencing. And now we really need tools to manipulate cells. And I think, probably the next decade to two will be like kind of in vivo cell engineering and reprogramming. None of this ex vivo crap where you have to engineer cells in complex manufacturing facilities and, allogenic stuff, like go directly into the body and make tools that can reprogram a cell or, reprogram a T cell directly or, transdifferentiate a cell. I think that's hopefully where I'm hoping we'll be at in 10 years, but we'll see.
So less electroporation.
Yeah, exactly.
Yeah. No, I think, that's really that kind of last comment about, so when you build these modalities, right, let's look before CRISPR at something like antibodies and like using them for oncology. That's based on both the science of antibodies, but also on the science of what are the signaling pathways in the development of cancer. So a tool often has its own biology, but there's the biology of the target. And as Omar said, the human genome was the biology that now basically 20 years ago has now been unlocked that we can target using CRISPR and using other genome editing tools. So really the question is, what's the biology today that we'll be able to unlock in the next 10 years? And you look at these huge efforts for understanding disease through the lens of cellular identity, cell states, cell types, what goes wrong and what changes during disease, and how do we modify between those cell states and types to cure a disease, potentially even before there's any sort of actual burden on the patient. Developing the tools that will connect to that biology is going to be the next wave of medicines, because now we're just not targeting at the gene level, it's at the cell level. And for as long as the genome and DNA has been a concept in biology, the cell is even much more ancient, hundreds of years more. So if we can find a way to programmatically control cells, like we're starting to with RNA or DNA, that will be the area that can unlock the next wave of therapies. And I think that's what we're most excited about, and how different tools can help us towards that vision.
I'm waiting for my personalized gene editing therapy. So I'll have to wait a little longer maybe, but I think that'd be really, really interesting, but maybe not so realistic in the next 5 to 10 years. But I think something that would be fun to do as we close, because I know you guys have stuff to run into and have to get back to it, but maybe some advice to young scientists. I saw this also, probably I read in Editing Humanity, but I read that Emmanuelle Charpentier said she got her worst grade in microbiology in school. And obviously we know that's what she specialized in. So, maybe I'll start. And my advice would be that don't put any sort of guardrails on what your potential is and what you can maybe achieve, and just dream the impossible. I'm sure when you guys were kids, you weren't like, I'm going to change the world and find new enzymes and create new therapeutics and new diagnostic tools. And you are. So my advice would be to, try to think big and achieve all the things that are possible. So just curious what your advice would be.
Yeah, I think those are great points. I think, one thing that can always be tough is trying to sort of break the mold. I remember when Jonathan and I were trying to stick together after grad school, a lot of people laughed at us and said, it's impossible in academia. You have to work alone and suffer through that pain. And so I remember we actually were talking to Eric Lander, of all people, about, the struggle of doing this. And I remember, he said, basically nonsense, sort of bend the metal frameworks of academia and, just, keep trying. Don't let people put you in a box, essentially. And he was like, that's what we did with the Broad. Like, it was unheard of, to build an institute like this and like be independent from the university and, basically build all these independent, units. What the Broad, not to go on off a tangent, right? But the Broad, basically built everything from scratch. And he basically bent, the framework of academia to do it. And I think that advice has always stuck with me because, I think doing what Jonathan and I is doing, it's not as common, right? There are instances of other people like at Berkeley, even at HMS that have done, joint labs. But I think, that process of being able to make that happen has been great. And so that would be my advice is don't let people put you in a box and, try hard to bend, I guess, the universe to your will to make what you want happen. So another thing I would probably also add is, if you're going to go through the path of science, like learn to enjoy the pain. That part is definitely true. I think, 99% of it is like looking at negative data and trying to iterate on that. And you definitely have to keep a strong mental sort of state if you really want to be able to keep going through all that. So that would be my second advice, is just, enjoy the journey and the process.
Yeah. Don't be afraid to fail. I think it's definitely an important one. I mean, I think Omar touches a bit, but I want to say explicitly, science is not a solo endeavor. It doesn't have to be, at least, and it shouldn't be, but science is a team effort and it's a community effort. So if you are inspired by something and you're interested in it, reach out to people. I think the one thing I didn't do when I was young is really find people who are super excited about science and work with them. And I think, the opportunity to do that with someone like Omar, you can see that works really well. The best scientists collaborate furiously, and they, build communities around them where you can pass ideas between each other. You can refine them. You can shoot things down, or you can say, I'm stuck here. Someone help me out. And, when you're maybe a younger scientist, you think that, oh, wow, I'm going to have to be alone counting worms under my microscope, or I'm going to have to compete with people in a science fair or something. But that's not really how it is. Science is about building relationships with, others, people who love this research. And it's about finding mentors who can show you how to do something. I mean, obviously, Omar and I are incredibly indebted to Fung for teaching us everything that he knows about how to do all of this genome editing and enzyme discovery. And then at the other end of that, it's about teaching mentees how this works and passing it down to the next generation. So if you love science and you want to get into science, one of the easiest ways to do that is find other people who have that infectious enthusiasm and just start talking to them, start collaborating with them, just pick their brains and let them pick yours. So I think that's always a nice thing to do. And they're now, thanks to the internet and all these communities, it's really easy. It's easier now than ever to do. But don't hesitate to reach out to other people and say, I don't know. And they can help teach you.
I love that, because I also think, everyone who's read the CRISPR story here, either, in the Walter Isaacson book or in Editing Humanity or others, you look at how many people were working on this kind of either subsequently, or, if they would have collaborated, maybe we could have had more. Maybe if the journals would have, allowed their papers earlier, this could have happened earlier. So there's so many steps that we can take to kind of make things better. But I think collaboration is a big one. We know that, having larger data sets helps make, stronger conclusions. And so, I think there's a lot of room for collaboration. And I'll certainly say that, even for me, I reached out to you guys. I read your paper on Cas7 and 11, and I was like, wow, these guys are geniuses. I want to understand more about RNA editing. And, both of you were so gracious and willing to explain the science. And so I think that definitely reaching out, there's a community of people who want to help and who want to make science accessible and interesting. So, thank you guys for that and for creating that, but also completely agree that it's, a community of amazing people who really do and want to collaborate. So I think that's awesome. And I think maybe I'll just say, is there anything that you guys want to touch upon as we close that maybe I didn't ask about? Anything you guys want to maybe spend a minute or two talking about?
Yeah, I just want to say, I'm constantly amazed by how sort of fast our progress to engineer cells and biology is accelerating. I think, I'm not even, as old as, of course, many of the other experts you've talked to that are in the field, right? But I feel like even in my time, probably a decade now in this space, I have seen such a flurry of new things and new technologies come out and new approaches to basically manipulate biology for the better. And I think, it'll be exciting to see what happens. But, there's, being in Kendall, it's just, you see so much, cool papers coming out every day, new companies coming out. The Bay Area, I'm sure, is very similar. Our office literally looks into Moderna and, being able to see, this past year, how fast we can basically come out with vaccines and updates for vaccines is, really exciting. And I think, at least in our own domain of being able to basically mine, right, molecular biology, mine, hundreds of thousands and millions of microbes for new proteins and studying those proteins and being able to find totally new biochemical activities that evolved probably over, hundreds of millions, if not billions of years that can then become useful for, health and society is also just amazes me every day. So, I think we're in the middle of like something special. At least this is, definitely the century of biology. And it's nice to sometimes just reflect on that.
Yeah. I mean, I couldn't have predicted like 5 years ago that this is what things would be like in terms of just, how much innovation is going on. And so I think that honestly, my predictions for the next 5 years are probably going to undershoot. So it's just kind of insane that, we get the opportunity to be working in this time, in this era when things are so, catalyzed for innovation. We were born too late to be able to, work on fundamental physics problems like in, quantum chemistry or something. But now we can be able to develop these tools. So I think that it's kind of unique to have that opportunity. We're just, we're lucky. But I think that we have to make the most of that and keep trying to, develop these medicines, these new technologies that can hopefully help people out. And that's really what it's all about.
Definitely. And I think you are definitely making the most of it already, with the Cas7 and 11 and all the other discoveries that have come out. And as you mentioned, you guys are just getting started. So many, many more things to come, and keep not letting people put you in a box. And I'm sure we'll see so much more from you guys. It's really exciting. So I'll be on the sidelines cheering.
Awesome.
Yeah, thanks so much.
Awesome.
ARC believes that the information presented is accurate and was obtained from sources that ARC believes to be reliable. However, ARC does not guarantee the accuracy or completeness of any information, and such information may be subject to change without notice from ARC. Historical results are not indications of future results. Certain of the statements contained in this podcast may be statements of future expectations and other forward-looking statements that are based on ARC's current views and assumptions, and involve known and unknown risks and uncertainties that could cause actual results, performance, or events to differ materially from those expressed or implied in such statements.